It has two, the skeletal and the cardiac muscles, one of which is the voluntary one, the skeletal muscles, and the involuntary one is the cardiac muscle. And then the smooth muscle is involuntary. So the smooth muscles are those that are in your intestines, mostly in the intestines, in the stomach. And the skeletal is the one in your arms, in your legs. That's the only one that's voluntary. Okay. So here, on the upper left of your image is the intestine. So the smooth muscle part is here. Our vas flexus, smooth muscle, serosa, and the mesothelium. And then this one here, this is the cardiac muscle. So as you can see, this is an individual muscle fiber. It is striated with the striations is the kanang pink lines no naa sa kaning lining the muscle. This is the nucleus of each muscle cell fiber, and then this black line that is dividing the muscle fiber is called the intercalated disc. Okay, and then this one is your skeletal muscle. So skeletal muscles, connective tissue investments, there are three: epimysium, perimysium, and endomysium. So it's the same as the one that we had in our connective tissue in the bone and the cartilage, the perichondrium. So that for the muscle, it's called mysium for muscle. Epimysium is the one that surrounds the entire muscle. The perimysium is the one that surrounds muscle bundles or fascicles or small bundles. And then the endomysium is the one that surrounds individual muscle cells. So epi is the entire muscle, perimysium is the small bundles, and then the endomysium is the individual muscle cells. So there. The entire skeletal muscle is covered by the epimysium. And then each of these, this is the muscle fascicle, this is covered by the perimysium, which contains the muscle fibers. And then the small dots here, it becomes like this. So individually, they are surrounded by the endomysium, which contains the myofibrils. So each myofibrils is surrounded by another covering called the sarcoplasmic reticulum. It consists of the sarcomeres, the Z lines. So this one, Monisha, so the sarcomere here in the lower right of the picture, this is the I band, the A band, and then this is the M line, the Z line, the titin. Okay, we will just discuss this one by one later. Okay, skeletal muscle cells. These are long cylindrical multinucleated cells. They have a sarcoplasm, a sarcolemma with T or the transverse tubules. And then the myofibrils has the thick and the thin filaments, the myosin and the actin, and the dark A bands and the light I bands bisected by Z disc. And then the desmin with plectin glues the Z disc together. So the dark A band, the light I band, and then thick filaments is the myosin. The thin filaments, the red thin filaments is the actin. So the thick ones, the blue ones are the myosin. And then they are, bisected by the Z disc, the light I band. So as you can see, the I bands here, this is a Z disc that bisects the I band. And then the one that glues the Z disc together is the desmin. So it looks like it's continuous because of the desmin, the zigzag line. Okay. So cross striations, the A bands is an isotropic that stays dark with thin and thick filaments. The I bands is the isotropic, lightly stained thin filaments, and then the H bands are light regions transecting A bands, also has thick filaments. And then the M lines, the narrow dark regions at the center of the H bands, which acts as the cross bridges. And then the Z disc or the lines, the dense regions bisecting the I band. Okay. You just go back to this picture. So the one that is not covered in the H band, these are light regions transecting the A band. And the M line, the narrow dark regions at the center of the H band. So H here is one, and then this is the M line transecting the H. Okay. Sarcomere is from Z to Z, so sarcomere is from Z to Z, from another Z line to another Z line, that is the whole entire sarcomere. So this is the functional unit of muscle contraction. They are covered by the sarcoplasmic reticulum. They are dilated terminal cisternae that encircle myofibrils at the junction of each A and I band. The function is to regulate muscle contraction by calcium. So sequestration, to relax, and then to release is when you contract your muscle. So releasing calcium, it means that you are contracting your muscle. So if you flex it, yung pagahiyon ng muscle, it's muscle contraction by the calcium in your sarcoplasmic reticulum. So the triad is a central T-tubule flanked by two terminal cisternae of the sarcoplasmic reticulum. They function to help provide uniform contraction. So that is the function of the triad. They provide uniform contraction. So if this is defective, so you can notice that some parts of your muscle is twitching, like it looks like pulsating, physically. So there is only a part of the muscle in your arm that feels like it's pulsatile. That is a malfunction of the triad, because this is the one that helps in the uniform contraction of your muscles. So again, sarcolemma, sarcoplasmic reticulum, the terminal cisternae, the T-tubule here, it's letter T, guys, like this. The white becomes T on the top in the sarcolemma. And then this is the triad. Here. 3, T-tubule flanked by two terminal cisternae. So, partially sandwiched, T-tubule and two terminal cisternae is the triad. This is an example of a skeletal muscle. So, this is, where is this? I think this is, I'm not mistaken, it's already near the stomach. Okay, this one on top is the, I think this is straight, yes. So it's not so clear, but what is demonstrated here is a striated skeletal muscle. So, as you can see, here, it is striated, and then the black dots here is the nucleus of each muscle fiber. There. So, just watch the video in the laboratory. It will be explained there further. So, for each muscle. filaments. The F-actin is a double helix arrangement with an active site for interacting with myosin. The tropomyosin is found in the groups of the F-actin helix, and then the troponins, troponin D, troponin C, and troponin I, they have different binding sites. The T binds to tropomyosin, the C has 4 binding sites for calcium, and then the I binds to actin, inhibiting the myosin-actin interaction. And then for the thick filaments, the myosin 2, they have 2 identical heavy chains and 2 parts of light chains. They are double-headed golf club, lobular heads, the S1 fragments of ATPase activity but require interaction with actin to release ADP and PI. Okay, the Huxley's sliding filament model during contraction, thick and thin filaments do not shorten but increase their overlap. The thin filaments slide past the thick filaments and then penetrate more deeply into the A band, which remains constant in length. And then the I and H bands shorten as the Z discs are drawn closer together. So this one. Initiation and regulation of contraction, depolarization plus calcium release will lead to actin-myosin binding, then it will result to muscle contraction. So, sarcolemma is depolarized in the muscular neurojunction. The T tubules convey depolarization to myofibrils, which is a voltage-sensitive dihydropyridine receptor. And then the calcium is released to the cytosol at A1 junction via calcium release channels at the junctional feet ryanodine receptors of the sarcoplasmic reticulum terminal cisternae opened by activated dihydropyridine receptors. And then sufficient calcium is released, and then it will be continued in the contraction cycle. So the activation of actin by calcium in the resting state, the myosin binding sites on actin are partially covered by the tropomyosin, and then the troponin I is also bound to the actin. So calcium plus the troponin C break the troponin I actin bond, and then tropomyosin shifts position and uncovers myosin binding sites, then becomes the active state. And then the relaxation, calcium concentration in the cytosol is reduced enough that the troponin C loses its bound to calcium. And then the sarcoplasmic reticulum, calcium pump binds calcium to the sarcoplasmic reticulum inner membrane with the calciquestrin. So medyo it is quite confusing, no, because it is the physiologic idea of how muscles contract and relax with the help of the actin, myosin, and the calcium in your sarcoplasmic reticulum. I will try to find a video that will help you understand it better, because it is more understandable if you see images explained during each event that is done during each process. So I will just send a link in our DCR with regards to how it is all done, and also the Huxley sliding filament theory. Okay. So the motor unit consists of the neuron and the muscle cell it innervates. A muscle may contract with varying degrees of strength because only some of the muscle cells contract. So the individual muscle cell is an all or none law. finer muscle movements require fewer muscles per motor unit, like the eye movement. So the innervation of the skeletal muscles, the motor nerve endings, and the sensory nerve endings in the muscle spindles and Golgi tendon organs for proprioception, and then the myoneural junction, the MNJ synapse, is a synapse between a branch of a motor nerve axon and a skeletal muscle cells. The depolarizing presynaptic membrane will go to the calcium channels to open, and then it will increase cytosolic calcium, and then acetylcholine is released, ACH, then they will bind to the acetylcholine receptors in the sarcolemma, thus depolarization again. So it's a cycle. So again, I'll try to give you a video on how these things work so that you can better understand. But okay, to make you understand, this is the axonal terminal of a motor neuron, the mitochondria, the synaptic cleft, the junctional folds of the sarcolemma at motor plate, and then this is part of the myofibril of the skeletal muscles. So this one here is the one, when the calcium channels open, they increase the calcium, this one. So when there is increase in cytosolic calcium, the calcium will get inside the axonal terminal of the motor neuron. It will then release acetylcholine, and then after that, the acetylcholine receptors in the sarcolemma will again depolarize. This is the one. So muna siya. The acetylcholine is released, and then they will bind to the sarcolemma again, and then the process again begins. Okay. Rigor mortis, it's a post-mortem rigidity appearing as hardening of the skeletal muscles. The body is dead, or in short, the skeletal muscles are already hardened, so it is rigid. So first few hours of death, it's not really that hard, but when the body will undergo rigor mortis, so the body is unable to synthesize the ATP. Although the myosin remains bound to actin, the muscles will just remain contracted. It will not relax anymore because nag-harden na ang skeletal muscles after death. So that is rigor mortis. Sensory receptors that monitor contractions. The first one is the muscle spindle. It provides feedback about changes in the muscle length and its rate of alteration. This one, the muscle spindle. And then the Golgi tendon organs monitor the tension and its rate produced during movement. So here, tension receptor organs. Okay, clinical considerations. The disease that involves the muscular tissue, the skeletal muscles, is the myasthenia gravis. It is an autoimmune disease in which the antibodies block the acetylcholine receptor. So once it is blocked, of course, there is no depolarization. So myasthenia gravis is more on stiffening of the muscles. And then second disease is the botulism, that is the food poisoning, in which the Clostridium botulinum toxin inhibits acetylcholine release at the myoneuronal junction. So, like the Clostridium botulinum is usually you can get it in spoiled foods, no? So it will release a toxin, and then if you have botulism, so you will just keep on vomiting because you have the muscles are they keep on contracting because the toxin inhibited the acetylcholine release. So your muscles in your stomach won't stop contracting. So that's the food poisoning part in your botulism. And then there are procedures known as the Botox. No, it's very common. The Botox cosmetic procedure. You use it to stop your muscles from contracting and relaxing at the same time. So it is always contracted mo nang if you have Botox, usually your muscles will stop moving. So if you can see the celebrities, no, if they have undergone Botox, usually it's very obvious because their cheekbones are no longer mobile, cannot move. Like it's like even if they smile, their cheeks remain stiff and smooth. So that's one of the examples of a Botox. So literally, a Botox toxin is inserted in the muscle, stop it from moving. So to stop also from aging, kay it will not move. It will stop from moving, so it will not cause energy release from the muscles and everything. So it will slow down also the age. So that's why some celebrities that are really old and then they undergo Botox, it looks like their face are cemented with something because it doesn't move. So that's the Botox cosmetic procedure. Okay. So for skeletal muscles, the shape and size of cell is long and cylindrical. The number and location of nucleus is many and is usually seen in the peripheral area. The striations is there. It contains striations. The T tubules and sarcoplasmic reticulum form triads at the A to I junctions, and then they have no gap junctions, and then there is a sarcomere, and the regeneration is restricted, and then it involves voluntary contraction. So it's the only one that is capable of voluntary contraction, and then distinctive characteristics is a peripheral nuclei. Okay, cardiac muscle. They contract spontaneously and display a rhythmic beat modified by the hormonal and neural parasympathetic and sympathetic stimuli. So they are poorly defined myofibrils. They have their calcium source is more of extracellular, and they have more mitochondria because of course it's an involuntary muscle that needs a lot of rhythmic beats to move. So it's the one responsible for pumping blood. So it has more mitochondria. And then the one that I showed you earlier, the intercalated disc, the black lines that are complex step-like junctions forming end-to-end attachment between adjacent cardiac muscle cells, and they are the functional syncytium. Okay, Purkinje fibers are modified cardiac fibers in the bundle of His, responsible for conduction. Okay, this is the cardiac muscle. So this is a normal cardiac muscle. Okay, so ang intercalated disc is this, the black lines in between this one, and then this one here, this one, and then this is the nucleus, individual nucleus. So the shape of cells are blunt-ended and branched. The nucleus is one or two and centrally located. Unlike the skeletal muscles, it's peripherally located. It's also striations, and then it is not a triad, but it is a dyad at the Z disc. Yes, there is gap junctions, unlike the skeletal muscles. It has no gap junctions. So the gap junctions in the cardiac muscle is the intercalated disc, and then there is a sarcomere. It is not capable of regeneration, It is not voluntary. Okay? Smooth muscle, it is non-striated. It is fusiform in shape, 20 micrometers in small blood vessels to 500 micrometers in a pregnant uterus. So, nucleus has a corkscrew shape during contraction, and then the sarcolemmal vesicles are called cavulae, and then contractile filaments, the actin, myosin, are not arranged in myofibrils because they are non-striated. And then the contraction is slower and longer. Thus, there is slower ATP hydrolysis. There is two types. The multi-unit which contracts independently of one another, and the unitary, single-unit smooth muscle. They cannot contract independently. So this one is this is the bladder, I think, yeah. So underneath the bladder is a smooth muscle here. It's non-striated. So here, shape and size of cells. They are short, spindle shape. They have single nucleus centrally located. They have no striations. The T-tubules are in the form of the cavulae and some sarcoplasmic reticulum. And then there is a gap junction called the sarcolemma, called the nexus, found in the sarcolemma. And then the sarcomere, they don't have sarcomere. They are capable of regeneration. They are not voluntary. And then the distinctive characteristics is the lack of striation. Okay, so do you have any questions? None? Okay. Let's proceed to nervous system. Nervous system overview. The CNS and the PNS, the central nervous system, and the peripheral nervous system. So the CNS contains the brain and the spinal cord, and then the PNS is the nerves that is outside the CNS and the associated ganglia. So they are functionally divided into two, into sensory and motor, somatic and autonomic for the motor. Then there's two types of cell, the nerve cells or the neurons which conduct impulses, and the glial cells or the neuroglial cells which support, nurture, and protect the neurons. Connective tissue investments, just like the muscle tissue, they have the epi, the peri, and the endo, only that it is neurium because it's neuro. Epineurium is a layer that is coating the external portion of the nerves. So it is a fibrous, dense connective tissue, fascia, that forms the external coat of the nerves. Perineurum surrounds each bundle of nerve fibers, and then the endoneurium is a thin layer of reticular fibers produced mainly by the Schwann cells that surrounds the individual nerve fibers. So the one producing endoneurium is the Schwann cells. They surround the individual nerve fibers. So same lang siya, no? For the skeletal and the nervous tissue, they have the epi, the peri, and the endo. Only lahi lang siya o sumpay. Mesonym ang sa muscles, neurium ang sa nervous system. So there, it's basically the same. This is the epineurium, and then the perineurium is the individual nerve muscle fascicle, and then the individual nerve muscle cell fiber is covered in endoneurium. Okay, the cells, neurons, contains a cell body, multiple dendrites, and a single axon. The morphologic classification is unipolar, bipolar, multipolar, and pseudo-unipolar. So the functional classification is according to if it is sensory, if it is an interneuron, or a motor in function. Neuroglial cells are the following the astrocytes, oligodendrocytes, Schwann cells, microglia, and the ependymal cells. So there, unipolar, bipolar, multipolar, and then the pyramidal, and these are individual types of neurons. So this is a typical neuron with its parts and synapses. Just recall this later. So this one is the cross-section of a spinal cord. You see it? So this one here is the butterfly shape, this one. This is the gray matter, this one. This one. Ng butterfly shape, and then this is the white matter outside. Sorry, white matter. This is the dorsal horn and the ventral horn. So dorsal horn, ventral horn is the spinal cord. That is a neuron, one in the pointer. So that is the dendrites, axon hillock, and the axon. Neurons, or the first one is the neuronal cell body. It contains the nucleus, cytoplasmic organelles, and inclusions, and the cytoskeletal components. It's also called the soma or the pericardion. So the nucleus is large, they are spherical in shape, and they are pale staining and is centrally located. They have abundant euchromatin and a large nucleolus known as the owl eye nucleus. Whereas the dendrites, their function is to receive stimuli from the sensory cells, the axons, and or other neurons, and convert these signals into small electrical impulses or action potentials that are transmitted toward the soma. So toward the neuronal cell body. They have arborized terminals, except in bipolar neurons. They lack Golgi complex in the cytoplasm, and then the organelles are reduced in number or absent near the terminals, except for the mitochondria, which are always abundant. The axons, they conduct impulses away. So the dendrites receive the stimuli. The axons, they conduct impulses away. So axon away from the soma to the axon terminals without any diminution in their strength. Some axons are as long as 100 centimeters. They originate from the axon hillock, which lacks the rough ER, the ribosomes, the Golgi cisternae, and the Nissl bodies, but contains many microtubules and neurofilaments. So the axoplasm lacks a Golgi complex, but contains the smooth ER, rough ER, and the elongated mitochondria. Axons terminate in many small branches, known as axon terminals, from which impulses are passed to another neuron or to other types of cells. Okay, neurons. Types of neurons. Melanin-containing granules, some neurons in the CNS, and dorsal root and sympathetic ganglia. The lipofuscin-containing granules, they increase in number with age. Lipid droplets usually are present. And then neurofilaments up to 10 nanometers in diameter are abundant and run throughout the soma cytoplasm with intermediate filaments. The microtubules is 24 nanometers in diameter and are also present in the soma cytoplasm. And then the microfilaments or the actin filaments is 6 nanometers in diameter, are associated with the plasma membrane. So glial cells are the ones that protect and support the neurons. The first one is the astrocyte. It's the largest of all the neuroglial cells. Its function is to scavenge the ions and debris from neuron metabolism and supply energy for metabolism. So there are two types: the protoplasmic astrocytes in the gray matter and the fibrous astrocytes in the white matter. Oligodendrocytes are the one needed for the survival of neurons in the CNS. They are responsible for producing the myelin sheath in the CNS. They have both the white and the gray matter. They possess a small, round, condensed nucleus and only a few short processes. There, oligodendrocytes. Schwann cells, they are flat cells with only a few mitochondria and a small Golgi region. They protect and insulate neurons by providing myelin sheath for the PNS. So the oligo is the one providing myelin sheath for the CNS. The Schwann cells are the one providing myelin sheath for the PNS. A single Schwann cell can only insulate a single axon, whereas a single oligodendrocyte may insulate several axons. Microglia, they are small phagocytic neuroglial cells that are derived from the mononuclear phagocytic cell population in the bone marrow. They have a condensed elongated nucleus and many short branching processes. Ependymal cells, or the epithelial cells, supply the neural tube and ventricles of the brain. They possess cilia which aid in moving the CSF, or the cerebrospinal fluid. Synapses. They are sites of functional apposition where signals are transmitted from one neuron to another or from a neuron to another type of cell. Classifications is according to the site of synaptic contact. So axodendritic, axosomatic, axoaxonic, and dendrodendritic synapses. Classification via method of signal transmission, so chemical synapse and electrical synapse. So chemical synapse, neurotransmission or neurotransmitter from neuron to neuron or neuron to muscle, delayed by 0.5 milliseconds. And then electrical synapse is done in the gap junctions, nearly instantaneous transmissions, and is less common. Here, common neurotransmitters. So, mostly are found in the CNS, and then only the acetylcholine is the one in the MNJ, and all parasympathetic synapses. So, excitatory, the glutamate, the dopamine, the aspartate. For pain suppression or inhibitory, the serotonin or the enkephalins and endorphins. And then the most common inhibitory neurotransmitter of the CNS is the gamma-aminobutyric acid, or the GABA. It's the most common inhibitory neurotransmitter. Okay. Nerve fibers, they are individual axons enveloped by a myelin sheath. No, it's not the same as the monosodium glutamate in the food, but it is those that are, you can have the MSG. It is also an excitatory for the CNS. That's why there are studies that are not, they do not recommend the MSG in the food because it will affect your CNS. But it is not the glutamate that is a neurotransmitter. Although it has glutamate, it is not B1 because naa siya sodium. It has a sodium with it. So that's why it's you can put it in the food, but it is too much MSG will affect your CNS. There are some studies about that before. That's why they banned the MSG for a while. But new studies now is that they are telling that the MSG is not harmful. Just use it in minimal amount lang. Do not use it excessively. Okay. So the nerve fibers, individual axons enveloped by a myelin sheath. The myelin sheath, again, is produced by oligodendrocytes in the CNS and Schwann cells in the PNS. They are not continuous along the length of the axon, but is interrupted by gaps called the nodes of Ranvier. And then they can be extracted by standard histological techniques. Methods using osmium tetroxide preserve the myelin sheath and stain it black. Nodes of Ranvier. And they are regions along the axon that lack myelin and represent discontinuities between adjacent Schwann cells or oligodendrocytes. So in the PNS, axon at the nodes of Ranvier is covered by a interdigitated cytoplasmic processes of adjacent Schwann cells that protect the myelin-free surface of the axon. Whereas in the CNS, it's found in the footplate of an astrocyte. So see the difference? So it is, they both have nodes of Ranvier only at different location. That one is a node of Ranvier. Murasha, it looks like an intercalated disc, the one in the pointer, but it is the one in your CNS and PNS. CNS. White matter contains mostly myelinated nerve fibers, but also some unmyelinated fibers and neuroglial cells. The gray matter contains the neuronal cell bodies, many unmyelinated fibers, and some myelinated fibers and neuroglial cells. The spinal cord gray matter appears in the shape of an H in cross-section of the spinal cord, which is the one that was flashed earlier with a small central canal and a ventral and a dorsal horn. So akong ibalik, I will go back to the picture of the spinal cord. So that one in the pointer is the central canal. And then again, the ventral horn and the dorsal horn. Brain gray matter is located in the periphery of the cerebrum and cerebellum, and then the white matter lies beneath the gray matter in these structures. The Purkinje cell layer or the is found in the cerebellar cortex only, consists of a flask-shaped Purkinje cell. They have a central nucleus, highly branched dendrites, and a single myelinated axon. And then they receive several 100,000 excitatory and inhibitory impulses to sort and integrate. So there. That is Purkinje cells with their dendrites, no kanang branching in the arrows. That's your dendritic trees. And then they are functioned to sort and integrate 1,000 excitatory and inhibitory impulses. So, as described, it has a central nucleus and highly branched dendrites with a single myelinated axon. So, the P is the Purkinje cell, the center of which the end of the arrow is the nucleus, centrally located. CSF, cerebrospinal fluid. It is a clear fluid produced primarily by the cells of the choroid plexus in the ventricles of the brain. So remember that CSF is produced by the cells of the choroid plexus in the ventricles of the brain. They circulate through the ventricles, the subarachnoid space, and the central canal, bathing and nourishing the brain and the spinal cord, and they also act as shock-absorbing cushion for the brain. They are 90% water and ions, and it contains just a little protein, occasional white blood cells, and infrequent desquamated cells. So in cases of meningitis, so that's why the cerebrospinal fluid is sampled via the spinal tap. Collect a sample of the CSF via your lumbar area in your spine. You collect it and then they test it for protein and for white blood cells, because if there is, and also they also test for sugar, because if there is an infection, whether be fungal or it is caused by tuberculosis, TB, meningitis, or the viral meningitis, those parameters, the protein, the white blood cells, and the sugar will increase or decrease depending on what is the cause. So that is why when they consider meningitis as the infection of the patient, they usually test the CSF, because they give a picture on what is your mode of management or what are the medications you need to give to the patient. So again, so to continue, they are produced and reabsorbed by the arachnoid granulations that transport it into the superior sagittal sinus. If the reabsorption is blocked, that's why there is hydrocephalus. So there is overproduction of CSF, and then there is no reabsorption, or the reabsorption is blocked. So if you have noticed, if a child usually has hydrocephalus, they put a shunt in the brain to drain the excess CSF, because what are the dreaded effects of hydrocephalus? So some of which is impaired mental capacity, and then some of which is more your brain will drown in fluid, no? So too much of the cushion na siya, it is not nourishing to the brain anymore if it's a lot of CSF fluid. So luoy ng mga bata na naay hydrocephalus. Nerve ending receptors, they are dendritic nerve endings located in the skin, the fascia, the muscle, joints, and tendons. They respond to stimuli related to touch, pressure, temperature, and pain. So the Meissner's corpuscle, nerve ending which are for light touch. They are found in the dermal papilla of the skin. And then for the Pacinian corpuscle is for the deep touch. They are found in the skin and the pancreas. So remember the two kinds of corpuscle. The first one is the Meissner's corpuscle. So they are responsible for light touch. For the Pacinian corpuscle, this one, it's like an onion skin, under the microscope. It's very pretty. It is responsible for the deep touch. It is usually located, this one is the pancreas. That is a Pacinian corpuscle. This one is a Meissner's corpuscle. So very distinct onion skin appearance. Okay, so that is the last slide for nervous tissue and muscle tissue. Okay. Do you have any questions? All right. So I will give you the quiz at 2:30. 10 minutes from now, I will open the quiz. I will close it at 3 o'clock. I will close it at 3 o'clock. everything is found in your notes, every question. And then for Thursday, that is your long quiz. That is also your study day for P1. So we will have no lectures on Thursday because on Friday will be your P1 exam. So be sure to attach your permits on the folder link that I posted earlier. And then the long exam on Thursday is 40 items. The P1 is 100 items for the lecture and 50 items for the laboratory. So the long exam on Thursday is at 7 a.m. Only for an hour. It's 40 items lang. And then for Friday, your P1, your lecture is for 2 hours, 8 a.m. to 10 a.m. And then your laboratory will follow 10 a.m. to 11 a.m. Okay. So for Friday also is the deadline for your lecture module and laboratory exercises. So just follow what is instructed in our GCR for the following topics. And then for those who are late enrollees, please catch up with your exercises from the beginning, your activity sheets from module 1 up to the present, because you already missed the initial quizzes that we had. So your source of points will be your module and your laboratory exercises for the first week. I cannot give you the quizzes anymore because it's unfair for those who attended the lecture last week and able to take the quiz on time. So I will not include it in the grade computation as well, the one that you're not yet enrolled to. But if you miss a quiz na you're already enrolled, then that's a different story, of course. So just for the new enrollees, just catch up with activity sheets in the lecture and the laboratory exercises. I think that's all for this afternoon. I'll just end this session and then just wait at 2:30. I will post the quiz. Thank you everyone for attending.
Thank you, Doc.
Thank you, Doc.
Thank you, Doc. Hi, Doc.
Salamat, Doc. Thank you, Doc.